Thank you for the introduction and the invitation. So I will talk about two related works from my PhD student, which was a collaboration between mathematicians, so Fabien Cros, my advisor, and me, and biologists from the École Normale Supérieure de Lyon and from the INSERM, Olivier Gandrion, Jacqueline Marvel, Christophe Arpin, and Xiaoying Wang. And I will start by introducing this schematic presentation of what is the immune response in the CD8 T-cells. So, CD8 T-cells are a family of lymphocytes that you can find before infection in some organs, like in the lymph nodes, in a quiescent state that is called the naive state. And when the organism is infected, the antigen-presenting cells, here in yellow, can present the antigen associated with the pathogen to the naive cells to activate them, and that initiates the immune response. So once activated, the CD8 T cells start to proliferate very quickly during this clonal expansion phase here, and they differentiate into what we call effector cells that are able to fight the infection by killing infected cells. And after a few days, most of those cells, approximately 90% of those cells, die during the contraction phase. And some of them differentiate in memory cells that will survive for months or for years in the organism to protect him more efficiently in case of new infection by the same pathogen. That's what we call the immunological memory. And eventually the second response if the same pathogen is encountered again. Okay, there is this interesting phenomenon called antigen-independent immune response, or autopilot immune response, that I will present through this experiment from Kaesh and Ahmed. They cultured in vitro naive CD8 T cells with antigen-presenting cells during a day. So the antigen-presenting cells actually activated the naive cells by presenting them the antigen. And at the end of the day, they removed the activated CD8 T cells, and injected them into a healthy mouse. So now you have cells that have met the antigen once, but that live in an organism without virus or bacteria. And yet, 6 days later, those CD8 T cells were effector proliferative cells, and 100 days later, they were quiescent memory cells. So that means that actually the infection is necessary to trigger the immune response by activating the CD8 T cells, but is no longer necessary for the immune response to continue. I mean, cells will differentiate, proliferate, die, etc., even if the infection is over. And that's a phenomenon that is not well understood, how this program of proliferation and differentiation can be encoded into the CD8 T cells, even in absence of a virus, and which is not reproduced by most of the mathematical models of the immune response. So I will talk about this later. And another important thing is that what I call the effector population is actually not homogeneous, and we can distinguish two subpopulations that are called the short-lived effector cells in red here, the SLEC, that are actually destined to die during the end of the response, while the MPEG for memory precursor effector cells actually contains the precursor of the memory cells, and then are destined to differentiate in memory cells and survive. And what you can just keep in mind for the following is that there is this protein, the protein TBET, which is expressed in CD8 T cells, and that is known to promote the development of the SLEC profile and to repress the development of the MPEG profile. So you can just keep in mind that through a gradient of TBET expression in CD8 T cells, we can use this gradient of expression in different CD8 T cells to characterize their their differentiation state, or yeah, if they are more SLAC-like or MPEG-like cells. And the important question is, what is the origin of this heterogeneity? And in this work, we considered the hypothesis that some heterogeneity can emerge upon cell division, which is a quite common hypothesis in biology. And more precisely, we suppose that it occurs because when a cell divides, its molecular content is not partitioned exactly symmetrically between the two daughter cells. That is an hypothesis that has been observed and studied in some references. And so it's like in this drawing, a mother cell divides and we get two daughter cells that can be slightly different from their mother. Okay, so let's start with this simple equation. Here, I denote by X the concentration of protein T-beta in one cell that is not dividing for now. So X is the concentration of protein T-beta, and we use this simple equation to model its evolution. The last term is a decay term with a constant rate delta. And the first term on the right-hand side accounts for a positive feedback loop between the protein T-beta and the gene that synthesizes the protein T-beta. So there is like an autocatalytic reaction on this gene. And then we model this with this Hill equation that is also used in a series of work from Friedman. So it's a quite simple equation that is known to be bistable if this relation of on the parameters is satisfied. So for any initial condition, well, we have two stable steady states, 0 and XS for stable, separated by an unstable one. And for any initial condition, we know what is the asymptotic behavior of the solution. And in terms of cell differentiation, we know if the cell is going, to become more a SLAC-like cell or MPEG-like cell. So now, let's introduce the phenomenon of molecular partitioning. I will use this notation. So X is the concentration, so number of proteins divided by the volume of the cell. Then the cell divides. We get two daughter cells, which are 2 times smaller than their mother, and inherits or more or less the half of the proteins. So if you compute the concentration in the two daughter cells, you get that those concentrations are 1 plus alpha times X and 1 minus alpha times X, where alpha is somewhere between, between -1 and 1. So in particular, alpha could be negative though. So 1 plus alpha couldn't be greater than 1 minus alpha. And so if we introduce this in the equation, we get the following system. Now we consider one cell that is dividing periodically. So the cell cycle length is supposed to be of fixed length that we denote by omega. Between two divisions, the concentration of T-beta evolves according to the bistable equation. And at each division, so at each time that is a multiple of the cell cycle length omega, the solution is multiplied by a factor 1 plus alpha k that can be chosen stochastically, for example, where alpha k is chosen in an interval that is included in -1, 1, and that more precisely is minus alpha max, where alpha max is just a parameter of the model that characterizes to which extent the division can be non-symmetric. The division of the proteins can be non-symmetric. So we studied possible behaviors of such an equation, and actually a family of equations like this one. And we get the results I will show just after that. Just to be clear, what is modeled by this equation is what we can observe on this drawing. For example, we start with one cell with concentration here, just above XU. The concentration evolves according to the bistable equation, then divides, so we get two cells, etc., etc. And the previous system is actually modeling just one branch of this tree. And if we want the whole tree, we can reconstruct this by adding a new equation each time a cell is dividing. So we are following one branch, and what can happen? So we can show that there are two interesting cases. The first case is if the molecular partitioning can be strongly asymmetric. By strongly, I mean that this interval minus alpha max is big, and big is bigger than this interval there. So precisely, there exists a critical value alpha star between -1 and 1, such that if the interval minus alpha max is bigger than the interval alpha star exponential of decay rates times length of the cell cycle -1, then for any initial condition, depending on the stochastic sequence alpha k, the solution can converges to 0 or diverges to infinity. So in other words, the solution can reach any positive value. So that will mean that the solutions, which are concentrations, are not bounded. And in terms of cell differentiation, that the differentiation is always reversible, because the concentration can go very low, then very high, then very low, depending on the stochasticity of the sequence alpha k. A second case is more interesting. Now we suppose that at each division, the partitioning of the molecular content is asymmetric, but not too much. And not too much means that this interval, in which the coefficient alpha k's are chosen, is small in the sense that it is included in this other interval. And then we can show the following property that I will just illustrate with a picture to avoid introducing many notations. So what you see here is a simulation. We start with one cell with concentration near the value xu that divides, and we get 2 cells, then 4 cells, then 8 cells, etc. And what you can show is that first, 0 is the steady state, which is obvious in the equation, and that you get what I call the stable region here, in the sense that in the solution enters in this stable area, it cannot escape the area, but it can take any value depending on the stochastic sequence alpha k. The solution can reach any value in this area, but it cannot escape. Around them, you can find the basins of attraction. So if a trajectory enters in one basin of attraction, then this solution will converge to the corresponding stable region or to the steady state 0. And in the middle, there is this region that I call undetermined fate, because if the solution is there, like for example at the initial time, you cannot predict what will happen. The solution can remain in this area forever, or it can escape it from the top or from the bottom, and if it does, then it enters in a basin of attraction and then converge either to the stable region or to the steady state. So in terms of biological interpretation, that will mean that if the heterogeneity generated at each division is not too large, then there exist 2 non-reversible states of differentiation, one with high concentration for the protein and one with low concentration for this protein. A last interesting result on this equation is that actually the longer the cell cycle, the thinner the red region here. So we know that during the immune response, cells actually are proliferating very quickly in the beginning of the response and then slow down progressively their cell cycle as the response evolves. And so if we consider now that the cell cycle is lengthening, so in this simulation, after each division, the cell cycle increases, then the undetermined region becomes thinner and thinner, and then you can see that at the end of the simulation, there is no longer cells with intermediate concentration. So that will mean that the lengthening of the cell cycle forces the cell to choose one direction or the other, to adopt one phenotype or the other, and not to remain in the intermediate state of differentiation. So now I will talk about a second model that actually is much more complex, that contains the previous one. It's a two-scale model with the cellular scale and molecular scale. At the cellular scale, we describe a discrete population of CD8 T-cells, that are the small cells here, and antigen-presenting cells, which are the big ones that can activate the small ones. Those cells, one cell is defined as a connected set of pixels on a two-dimensional domain, and those cells evolve according to stochastic movement laws. They can interact together. There are quite a lot of rules, so I will be quick. And depending on what is happening on the second scale, the molecular scale, those cells will divide or differentiate or die. Okay. So the second scale, so is the molecular scale. And here we consider this quite complex, but actually very simplified molecular regulatory network that contains a few genes or proteins that are important to describe the properties of a cell, the properties that we are interested into. Like, for example, the ability of a cell to interact with its environment. Just for example, those are receptors for a protein that is called IL-2 that can diffuse in the domain. So the cells can sense the IL-2 in the environment through those receptors. They can interact with the antigen through these receptors, etc. I won't go into the detail because my talk is too short, but if I can say, this network is kind of translated into this system of 6 ODs that models the concentration of 6 proteins in a CD8 T-cell. I just want to focus on the third equation, which actually is exactly the same as in the previous model, except for the first term here, lambda T1 FCPA. But actually FCPA is just a function that is equals to 1 when the CD8 T-cells meet an antigen and 0 overrides. So actually when the cell is activated, if there is no more antigen in the domain, then this term is 0 and we get exactly the same equation as in the previous part. Okay. And yeah, still at the molecular level, we use this PD to model the secretion of one protein, the protein IL-2, which is associated with cell activation and cell proliferation, that then diffuses on the two-dimensional grid. So I have not time to comment this properly. And finally, to finish the presentation of the model, this is the kind of differentiation scheme we use in this model, which is based on thresholds. There is a system of thresholds. For example, LR here is the concentration for the activated form of the receptor for the protein IL-2. And we say that if this concentration crosses a fixed threshold, then the cell becomes activated and starts to proliferate. There is a threshold on the protein Tibet so that the cell can become effector, a threshold on the protein EOMES so that the cell can become a memory cell and stop proliferating. And finally, there is a threshold on another family of protein, the protein caspase, so that if this concentration crosses, this threshold, then the cell dies immediately. Okay, I will show you a few results of simulations. So here what you can see is, just before I comment this figure, I need to say that, as you can imagine, this model is very time-consuming to compute. So we couldn't perform a proper parameter estimation, etc., a proper mathematical analysis. So what we did is to find as much as possible parameters in the literature. Some parameters have been estimated on previous versions of this model developed by 2 postdocs, Sotiris Procopiou and Shefang Gao. Some parameters have been fixed at reasonable physiological values. And for the other ones, we just compute a huge collection of set parameters and choose the better one. Based on in vivo data at both the cellular and the molecular scale. So what you can see here is the total number of cells normalized by the number of cells at day 7. So the full line is for the simulations and the squares are for the in vivo data. In black is the first immune response, so naive cells are activated, then expand, contract, etc. And the blue lines are for the secondary immune response, so we took the memory cells generated at the end of the immune response and activated them again with the antigen. We can also look at the composition of the population in terms of fraction of cells in each phenotype. For example, in gray, it's the fraction of naive cells. So we start with only naive cells that are activated. And then we see that at day 6, the whole population is composed of effector cells that progressively decrease as the fraction of memory cell increases. We have had also data at the molecular scale. So what we had is the mean expression value for 3 proteins. Protein IL-2 receptor that are activated that are associated with cell activation. So those receptors are highly regulated at the beginning of the response and then controlled at low level. The protein TB8, which is associated with effector phenotype, and the last one associated with the memory phenotype. You can see that the last time point is not in good agreement between the model and the in vivo data. So maybe that something is missing the model to control this expression at the end of the response. But this time point is also surprising compared with what we can read in the literature. So maybe that it could be nice to have another time point to be sure that this decrease is real. And actually, it's not very interesting to look at the mean expression value of proteins to understand what is going on. So I will finish with this simulation result where we can see the concentration of those 3 proteins in each cell of the population. So this is the result at day 5 after infection. Each column corresponds to 1 cell, and for each cell you can read on the y-axis the concentration of 3 proteins, protein TB8 in red, EOMS in blue, which is associated with memory, and caspases in brown, associated with this. Those cells are ranked from the one with the lowest concentration of TB8 to the one with the highest concentration. So what we see day 5 is that the population is heterogeneous, but this heterogeneity is quite it seems to follow like a uniform law because the concentration is like a straight line. Day 8 after infection, we see that we have more cells and the distribution is quite the same. But progressively, we see that we have 2 subpopulations that are emerging. On the right, a population with high concentration of caspases in brown. So those cells are actually dying progressively. That's why the contraction of the population is starting. And on the other side, we see cells with high concentration of the blue protein EOMS. So actually, those cells are becoming progressively memory cells and stopping proliferating. So I skip a few days. It's the same dynamics. And you can see that at the end of the response, we have 2 clearly distinct subpopulations. One on the right, which is actually progressively dying, and one composed of memory cells that are not proliferating anymore. So I will conclude. With this model, we propose an explanation for what I call the Autopilot immune response, where we have 2 subpopulations of cells with opposite phenotypes, and a contraction of the population, so a decrease in the number of cells that start when we have a lack of cells in intermediary state of differentiation. And actually, the study of the equation from the first part, the impulsive equation, shows that those non-reversible phenotypes, we have non-reversible phenotypes only if the heterogeneity due to stochasticity, if the stochasticity is not too strong, and more that the lengthening of the cell cycle can be an argument to precipitate the differentiation of cells toward one of the other lineage. Okay, and this is a remark about the model complexity that we should reduce, but I am over, so I stop there. Thank you.
Okay, I was just one quick question. I was wondering how many of the parameters do you actually can calibrate with the real value, experimental values?
I don't remember the exact number because some of the free parameters have been fixed with physiological values by hand, but the real free parameters that we have tested, I think something between 13 and 16, I guess. Between 13 and 16.
Okay. And how many parameters do you have in total?
For the I don't know. For the molecular scale, something like between 20 and 25, I guess. And for the cellular scale, there are many parameters but most of them have been calibrated on a simpler version of the model previously, though, so they were already estimated.
But okay. And so, okay, so what method do you use if you have so many parameters, you know, to feed them to experimental data?
It was a huge problem. So we just selected ranges, possible ranges for those parameters, then generated a huge collection of parameter sets in those regions, simulated them, took the better one, to have a thinner grid and did it again a few times. Okay. So, but I know that that doesn't prove that the identifiability of the model that definitely need to be simplified. That was the last part I didn't talk about in my conclusion.
Okay, thank you.
So there's someone who's asking if you have access to measurement of the heterogeneity.
Of the heterogeneity? That's very difficult because there exist experiments with individual expression for different proteins, but usually what you measure is the fluorescence of a cell, and you don't. It's not easy to compare directly the fluorescence to the concentration of a protein in different cells, and the fluorescence cannot be the same for different proteins. It's very difficult to compare those figures actually with the model.
Yeah, like single cell, I don't know.
Single cells, yeah.
Yeah. So for single cells, you wouldn't be able to measure, even if you get the single cell data, you wouldn't have single cell data for
qualitatively, for sure. Quantitatively, what is possible is to make a rescaling from, because the unit, the physical, we don't measure those physical quantities with the same unit, so we have to rescale it. Maybe that we can compare it, but I don't. I search a lot for such data. That's not so easy to find dynamics data for this thing. And I don't know, the answer was that we couldn't compare it. I cannot remember precisely what were all the arguments for that, but it seems to be more complicated than that.
Okay, thank you.
Yeah.
Okay, if there are no other questions, maybe we can continue the discussions at the break. So thank you again, Simon.